Welcome to our series. Our team is grateful you are here. Of course, seeing you in person would be preferable, but given the ongoing response to managing the SARS-CoV-2 pandemic at Hopkins and in our own local communities, these virtual platforms have proven efficient for sharing information and for education and discussion. A couple of updates. This is our first session of the fall semester. Please continue to review our offerings throughout the fall. We'll be adding some content as well as to include a novel time to share questions and insights with our clinical deputy director of the Institute, Dr. Noah Hahn. Our team is working on building this out and we'll have updates shortly. Please keep connected. You can find our updated programming and our newsletter on the web. Just Google Greenberg Bladder Cancer Institute. And I wanted to again draw your attention to our group of educational supportive care programs, specifically tailored around the experiences of women with bladder cancer, developed and led by Doctors Armine Smith and Jeannie Hoffman-Sensis, and the program team of Pamela Getz, Samantha Rockler, and Stephanie Cooper Greenberg. Again, all of these are up on our site, and if we can help you directly, we are only an email away at bladdercancer@jhmi.edu. I'm grateful to all of you for joining us today for a conversation about metastasis, the process when cancer spreads. I'm grateful that a key collaborator for our institute, whose partnership has been important to the ongoing growth of our research programs, has joined us today. I hope you will allow me the chance to introduce you more formally to Dr. Andy Ewald. Dr. Ewald is director of the Department of Cell Biology at the Johns Hopkins University School of Medicine. He co-leads the Cancer Invasion and Metastasis program in the Kimmel Cancer Center. He is professor of cell biology, biomedical engineering, and oncology, and his work seeks to understand how cells build organs and how, when hijacked, these same processes can contribute to cancer metastasis. Dr. Ewald received his undergraduate degree in physics with honors from Haverford College. He earned his PhD in biochemistry and molecular physics from the California Institute of Technology, also known as Caltech. He joined the Johns Hopkins faculty in 2008. He's an active member of the American Association for Cancer Research, the Society for Developmental Biology, and the American Society for Cell Biology. His work was recognized with the 2011 Morphological Sciences Award from the American Association of Anatomists for his contributions to the field of epithelial morphogenesis. Andy has agreed to start us off by speaking about his work, and we will jump to a larger group discussion. I would ask for attendees to bring questions into the chat, and after Andy's presentation, we will have ample time for group discussion.
Thank you, David, for the very generous introduction and for the opportunity to talk with your group today. Let me share my screen. All right, I believe the slides are now showing. I want to start by thanking my laboratory, the people who actually do the work. I have to figure out what we should do and how to fund it and how to communicate it. And these are the people who actually do the work. So, get rid of that. We're interested in metastasis because it is the major driver of deaths from cancer. This is schematized in, or easily understood in breast cancer, where local breast cancer, where all the cancer cells are confined inside the breast, has extremely positive patient outcomes because modern trimodal therapy, so surgery, radiation, and chemotherapy, is able to eliminate cancer from the bodies of these women in almost every case. However, when the cancer cells have acquired the ability to spread and grow in distant vital organs, 5-year survival rate is much lower. This is where most of the suffering and most of the death occurs from cancer across organ sites. So there's really two questions involved. First is, how does it work? And the second is, how do we stop it? So it's the major clinical problem across breast cancer because it's driving patient outcomes. However, metastasis remains incompletely understood. I wish I was coming to tell you that we understand how, in great molecular detail, how every step works, but it's not completely understood. Current therapies are not effective enough, and I'm also here to tell you that research is the only way to change that. If we want to improve patient outcomes, we've got to have more basic research to figure out how metastasis works and how we can more effectively treat it. So it's important, but it turns out it's also hard to study, right? Why is it we don't understand it completely? Well, it's in part because it occurs deep inside the body, right? You're thinking about cancer cells in the breast or the bladder, accessing the circulation, spreading through the body, getting to a distant organ. There's dynamics involved from seconds, right? Individual molecular interactions to decades, which is more the time it takes for a cancer to originate, spread through the body, and grow to dangerous size in a distant organ. There's also a challenge because tumors are highly complex. There's thousands of changes that occur from when a normal epithelial cell goes to becoming a metastatic cancer cell. And it's difficult to understand where to start, right? We need actually systematic ways to think about how to identify what's important because, sorry, that just jumped on me. The biology is changing at the molecular, cell, tissue, organ, and organism level. So we need to bring together the right expertise to be able to work across those different types of biology and to be able to identify what's most important to study. All of these challenges are shared with the study of normal organ development. So if we want to understand how the normal breast forms, we have these same challenges that occurs deep inside the body. The dynamics take place over multiple timescales. The organ's highly complex, the biology changes. So I actually first got into the study of the mammary gland, trying to understand how it was built in the normal context. And what you can see here is two examples. We see a 3-week-old mouse mammary gland. You see the organ is pretty close to adult size, but the network of ducts, these are the epithelial tubes that will make and carry the milk during lactation, are just rudimentary. There's a very small number. There's very simple design. By 10 weeks of age, which is the end of puberty in mice, right, we see a large complex network of epithelial ducts. So this turned out to be, for me, an outstanding context in which to understand and quite literally develop the toolkit to model complex tissue and organ biology. So I'll show you a couple simple examples of that and then get back to metastasis research. So if you've heard about scientists working on cancer research, you've probably heard about them working with cell culture. What that typically means is something like what you see at the bottom there, where there's a petri dish. There's a set of cells that are growing on the bottom surface, that piece of plastic, with media, so a liquid media that feeds them sitting above them. And you can put virtually any type of cells in there. So you could harvest cells from the normal mammary gland, isolate the epithelial cells, and then put them in this cell culture. The question is, will they do what we need them to do? Will they make a network of epithelial tubes that helps us to understand how the breast forms? And the answer is no. If we take mammary epithelial cells, which you can see here running around on the surface of this cover slip, if you put them in 2D culture, they don't make tubes. And if we start with cancer cells, they don't make tumors. Instead, you get a bunch of cells running around in a plastic dish. There's a lot of features inside the cell that can be effectively studied here, but the interactions among cells and the generation of tissue architecture cannot be studied there. So I got into this field, but with a relatively simple observation. It wasn't mine, it was other people's. It was Ewald, Mina Bissell, among others, had argued that there was something really important you needed to have if you wanted to study how tissues formed, not just how cells behave. And that's that you needed the cells and you needed their normal 3-dimensional environment, which is composed of a meshwork of proteins collectively referred to as the extracellular matrix. So their argument was if you take the organ and you get the right cells out, you put it in a dish, plastic dish, right? Of course, it's not going to look like the organ, but you could get surprisingly close if you just take those same cells and put them in a 3D gel of extracellular matrix or ECM proteins. I want you to think of this like putting raisins in Jell-O, right? They're floating inside that Jell-O mold. And it's actually kind of surprising how big a difference it makes. This is that same sort of cells, and instead of putting them on the surface of a cover slip, we put them inside this gel, and you see that they form a branched network of epithelial tubes. We get thousands of these structures for normal mouse, hundreds of thousands per breast tumor, right? So we're able to study this at very significant scale. So we don't have to just look at one at a time. We actually culture thousands per dish, and we can go back and forth with robotic microscopes, as we did here. This was one of several hundred movies collected at the same time over a period of days to understand what's changing. When we do that, when we take cells out of out of an organ or out of the tumor, and we put them in this 3D gel of extracellular matrix proteins. That's frequently referred to as organoid culture, right? You've probably heard this word before: organoids, bladder cancer organoids, breast cancer organoids, tumor organoids. And it really is as simple as the word suggests. Organ, right? And oid just means like the organ. So it's a set of culture conditions, which is subtly different for each organ, for each tumor type, right? That allows you to grow the cells of that organ in a way that they look like the architecture of the tumor in vivo, of the normal organ in vivo. So broader context, these are live cultures of mammalian cells. They're grown in a 3D protein environment. And it's very important that it's a technique for amplifying rare tissue samples. So if you start with a very small sample, say a biopsy of a tumor, and you put it in these 3D extracellular matrix gels, the cells will divide and grow, and then can be split and then divide and grow, and then can be split and will divide and grow. And you can then generate a large amount of material to study from a small amount of starting material. This is very important when you're working with human tumors. Organoids can be used to test new therapeutic concepts, perform genetic experiments at a large scale, and image cell and molecular dynamics. I can show you 3 examples here where you're looking at, in this case, all of the nuclei and all of the cell membranes in a mammary epithelial duct as it's elongating. Or we can focus on the behavior of a single cell within that epithelium as it pushes the duct forward. Or we can look at a major oncogenic signaling pathway, the PI3 kinase pathway, in an individual cell in that duct as it's elongating. We're actually able to watch the molecule's activity in 3 dimensions over time inside the cell. So this convinced me, working with these 3 different contexts, convinced me that it was actually a very powerful approach for studying mammalian biology. So I got hooked. We made some significant contributions to the study of normal memory development, but now most of our effort in the lab, about 10 to 1, goes to understanding metastasis. I want you to think of metastasis as being a multi-step process. It's not just the end result that there's a tumor at the other end, in the lungs or the brain or the liver, right? It's a multi-step process. Think about it from the point of view of the cancer cell. The cancer cells divide to drive tumor growth, but that's still a local tumor, right? That could potentially be surgically cured. Eventually, to metastasize, they need to invade into the surrounding tissue, travel through the blood, escape the immune cells circulating in the blood, arrive in the distant organs, and then overcome a bunch of intrinsic limitations to divide quickly enough to form clinically significant tumors. We'd like to understand this. We'd like to treat it better. And I love to show this slide because it highlights that these tumors don't come annotated with explanations. What you're looking at here is a breast tumor that's been fixed and sectioned. So think of it like a deli slicer. If you go to the deli, there's, say, some cheese, they put it on the deli slicer, able to cut very thin slices of cheese. What you're looking at here is a very thin slice of a tumor. And you can see the dark dots there, dark purple dots are the nuclei. These pink smears are immune cells coming inside the tumor. The big circles are the adipocytes, or the fat cells, and the red over here is muscle, right? So you can see that there's just a tremendous complexity among the different cancer cells. Some are round and light, some are dark and spindly and sort of drawn out shapes, right? It's not clear who's who, who's doing what, and what's even happening, let alone what we should be stopping or how. And so we've taken an approach across a number of years now, chiefly in breast cancer, but increasingly across solid cancers, that we try to understand how tumors work by taking them apart into their constituents, and then we test whether and how each of those parts contributes to metastasis. We then try to understand which of those parts are essential and could potentially be targeted with drugs. The guiding principle here is that we're trying to let the cells tell us which factors are important. We don't want it to be my own philosophy or models or big ideas. We want the cells to teach us what's important and what we should be targeting. So I showed you normal breast tissue a couple minutes ago, growing in 3D in Matrigel in the laboratory. This is actually a breast tumor, right? So this is from a patient when I was at UCSF. This was from a patient from the clinic there, and the patient was diagnosed with stage 3C breast cancer. 20 lymph nodes were biopsied. All 20 lymph nodes showed evidence of cancer cells growing in them. So this is a tumor that in that patient was spreading vigorously through the body. Yet, when we grew it in culture in the laboratory, it stayed very confined. you'll notice nice smooth epithelial edges around this tissue, and you notice an absence of cells out in the extracellular matrix. I promised we looked at higher magnification. We used different ways of staining to highlight the cells. We didn't miss them. This was a human breast tumor organoid from a patient with a highly infiltrating, highly disseminative tumor. And when we put it in this 3D environment, the cells just didn't invade, which led us to realize that there might be something missing in the way we were studying those cells, some critical co-conspirator in the environment around the tumor that was critical to their invasive behavior. So we thought, okay, maybe something's missing in our assays. Could it be the extracellular matrix, right? And so what you're looking at on a slide here is a very different stain. Now in dark sort of brown is the breast cancer, the breast tumor with the cancer cells. And in gold shiny signal is the fibrillar collagen 1. Collagen 1 is a particular protein of the extracellular matrix on which cancer cells are known to invade. And so we hypothesized that collagen 1 in the tumor microenvironment was really important to the invasive behavior of the cancer cells. But I want to be fair, lots of other people were putting a green arrow over here, right, and saying, okay, it's something in the tumor microenvironment, but it was something different. They would say, okay, it's the fibroblasts, or it's the lack of oxygen, or it's the presence of a certain immune cell. So what I want to highlight as well about this organoid approach or 3D culture approach is that we can take what are very difficult questions to answer in a patient, and we can pose them very precisely and answer them very definitively in 3D culture. So which parts of a human breast tumor are most important? Well, we can take human breast tumor organoids from the same patient and put them into that gel that I was just showing you that has proteins like those that surround a normal breast duct. And tissue and organoids from that same patient can be explanted into a gel composed of proteins that surround a breast tumor. If the extracellular matrix isn't important, right, the left and the right movie you're going to see next should look the same or pretty close, right? In contrast, if the extracellular matrix is an important regulator of cancer cell behavior, the movies in the next slide should look very different. These organoids were isolated from the same patient, explanted into 2 different ECM gels, imaged at the same time with the same media. The only difference here is there's proteins in the tumor microenvironment. Let's start that over. It's kind of an interesting pair. And you'll notice that the cancer cells grown in that normal-like environment remain very confined. There's a really nice edge to the tissue, and you don't see cells escaping. In contrast, when they're in that 3D extracellular matrix environment, like that around a breast tumor, composed of collagen 1, they invade vigorously away, they escape. So we published some nice papers on that. We gave that, we were able to establish the concept in breast that's since been validated in multiple other tumors, that the environment around the breast tumor was really important, right, for regulating whether it could metastasize or not. But a couple of years later, we realized we could use the same experiments, but flip the logic, flip how we thought about the experiment, and force the cancer cells to tell us which genes are most important for invasion. So if you look here, you see one of these organoids has these clear invasive strands. And the basic idea we had was that the cells that are actually invading, the tip of the spear, if you will, coming out from this small piece of tumor, was probably more specialized for invasion than the cells that are inside the bulk, right? I want you to think of this like the Boston Marathon. If you go to the starting line of the Boston Marathon, you'll see the elite runners. They all want to win. They're all really fast, right? They're all wearing running gear. And it's really difficult to predict which of them are going to win that day, unless you know a lot about their past performance. In contrast, if you go to mile 26.1 of the Boston Marathon, you can predict the winner of the Boston Marathon because it's the guy in the front, right? He's about to cross the finish line. This is the same idea. We can predict which cells are best at invasion by just putting them in an invasion-promoting environment, looking at the cells at the front and seeing if they're different. And what we're guided here is that there's different cell types in the mammary epithelium. In fact, really, I should talk about it as 2 different compartments. There's a basal compartment, which sits on the outside, and a luminal compartment, which sits on the inside. I realize that the colors are switched in the title. Please look at the colors in the bottom here. So the inside, right, the luminal compartment is keratin 8, keratin 18, and E-cadherin. These are just 3 different genes. The outside is keratin 5, 14, P-cadherin, and P63. Again, different genes. They're just ways for us to label and keep track of luminal and basal. Bladder cancer has the same luminal and basal dichotomy because the bladder has the same luminal basal dichotomy in its normal state. So the question here was super simple. Is there anything different about the cancer cells leading invasion at the molecular level? And what we found was that there was. The cancer cells at the front expressed a set of genes related to basal epithelial differentiation, like keratin 5 and 14. And they need these specific tools in order to invade and metastasize. So there was differences between the cancer leader cells from the bulk. And there was these genes code for tools that we shown were important for metastasis. If you blocked access to those tools, the cancer cells stayed in the primary tumor. What was really interesting is that different types of cancer cells reuse the same tools. This isn't the only mechanism of metastasis. I'll tell you about a different one we discovered recently, but this is a conserved mechanism where you see different cancer types of luminal A, that's the least aggressive, luminal B, moderately aggressive, but still pretty differentiated, and triple negative breast cancer all have very different patterns of invasion. But the cells leading invasion express keratin 14 in all three cases. So different types of breast cancer cells are activating similar genes to escape the tumor. We're trying to understand the tools they're using and which of those can be targeted with drugs. Okay, so genes, I want you to think of as coding for the instructions, right? It's like the library. Somewhere in the library, there's instructions to make of the cell, right? Inside the nucleus, there's instructions to make anything a cell could need to make. But in order to act on that, you need to make RNA, right, which is essentially the blueprints being copied out from the library and then handed off to the job site, where then you can go ahead and make it into a protein, right? So the genes provide the tools that allow cancer cells to escape. So we wanted to, again, flip the logic. And instead of thinking about the microenvironment tricking cells into leaving, we wanted to put the cells in that suppressive microenvironment, that normal differentiating microenvironment, and change something inside the cell, right, inside this blue cell that would cause it to migrate away. And there was a specific gene that we thought might be very important to this because it's what's called a transcription factor. That means that it's able to control the regulation of many other genes. So you turn this gene on, hundreds to thousands of genes also change, right? So its name happens to be twist1. So we want to test, is this gene important? This is another way of doing that parts list comparison, right? I think it might be important. How do I test that? And what we do here is we take tissue from the same epithelium and we either leave the gene off or we turn it on, right? And we're able to test whether it's important. If the gene does nothing, the two movies should look the same. And both are being cultured in that suppressive matrigel environment. So I hope you can appreciate that on the left, the control cells are differentiating and branching in a very normal way. They're forming new epithelial ducts. On the right, it's getting pretty crazy, right? What you're seeing instead is that individual cells are leaving that epithelial group, migrating away and forming new sites. If you look on the left here, there's new sites forming next to that epithelium. Interestingly, we didn't change anything else. This still isn't really a tumor, right? It's a normal epithelium expressing one gene. What's really nice, though, is we control exactly when that gene turns on by addition of a small molecule to the media. So we can then go and measure what changes when we turn the gene on. I told you it was a transcription factor. Well, it turns up 107 genes to a high level, and it turns down 76 genes to a low level, right? It's easier to target the increases. So we focused over there. And what we checked was whether inhibiting a single twist1 target, something called protein kinase D1, would affect cancer invasion. And we found that it did. Blocking this one target, either genetically or with a small molecule drug, blocked invasion and metastasis in mouse models. And even more excitingly, when you look at human breast tumors, we saw the same effect, that the vehicle, so if we just leave it untreated, we get extensive invasion. And whether it's a mouse model or a human breast tumor grown in culture, blocking this one kinase was sufficient to block invasion and dissemination. So I think it's an example of how you can take a very simple and controlled experimental approach to understand how a single gene turns on a series of other genes and then find targets for potential therapies. We can also study how the different tissue pieces interact, right? So if for a cancer cell to leave the primary tumor and get to the lungs, it's going to need to at some point enter the blood vessel. And we thought that was a story about cancer cells detaching, leaving the tumor, migrating through the ECM, and entering a blood vessel and flowing away. So we wanted to test that model working with the material science department, Peter Searson in the material science department. So we made a microfluidic device. That just means we can control the movement of small amounts of liquid and the placement of cells. So we were able to put cancer cells in one location and a functional blood vessel in the other, and then image what happened. And what we saw surprised us very much, surprised the community, which is that the tumor simply puts a foot down into the edge of the blood vessel and then expands its footprint. And when you come back, part of the vessel wall has been completely replaced with cancer cells. When you look at this over a longer period of time, you see that a relatively innocuous group of cancer cells can approach the vessel wall, replace the vessel wall, and then release cancer cells into the circulation. So we concluded in this study that cancer cells can rapidly integrate into vessel walls or obstruct the flow of blood in these vessels. Cancer cells and endothelial cells form tight junctions with each other, so they don't leak the blood or the sera. It's actually surprising how well the cancer cells can substitute for blood vessels. Sorry, I don't know why this is jumping. And these are called mosaic vessels because it's a mixture of blood vessel cells and cancer cells composing the blood vessel. And they're actually frequently observed both in mouse and human tumors. We've done that work in breast, collaborators have done that work in pancreas. And this direct release into the circulation could actually account for a high fraction of metastasizing cells. Currently, we're putting a lot of emphasis on something called triple negative breast cancer. It's called triple negative because it lacks expression of estrogen receptor, progesterone receptor, or HER2, which are the really main axes of therapy in breast cancer. Since these cancer cells don't express those, endocrine therapy and HER2 or targeted therapy like Herceptin doesn't help these patients. So we're really focusing on this disease both because it has an aggressive natural history and because there's a limited number of therapeutic options. So what we found was that triple negative breast cancer actually uses very different tools to invade and metastasize than luminal breast cancer. On the left, you see a luminal breast cancer organoid, and you see that it's expressing this adhesion gene, ecadherin. You're seeing all the nuclei with DAPI. You see it has these big, bulky invasion patterns, invasion strands that are basically not green, right? They're not expressing this other gene called momentum. You look at triple negative breast cancer, whether you look at mouse models or human tumors propagated in mice, or you look at fresh human tumors cultured in the laboratory, right? You see that the cells at the front are expressing vimentin and ecadherin, what's called a hybrid epithelial to mesenchymal transition. I want you to just think of it that they're stealing the best features of both an epithelial cell and a mesenchymal cell like a fibroblast and mashing them together in a way that makes them particularly dangerous, right? And I don't understand why that's jumping. We then used single cell sequencing. What that means is we harvested single cells, and then we measured every single gene that's being expressed in those single cells across thousands of examples to understand how these cancer cells change their equipment as they metastasize. And we identified control genes that control invasion and metastatic colony formation, and an unexpected diversity of cell states in the triple negative breast cancer metastasis. So basically, as the cancer cells got to the distant organs, they changed into more flavors of cancer cells, which actually makes it particularly difficult to design effective treatments. Fortunately, there are still therapies that they are sensitive to. That also led us then to try to look at what was different inside a tumor, right? So if we have a single example of a triple negative breast cancer tumor, right, and we put 1,000 organoids in culture, it turns out they don't all behave the same. I'll deliberately restart that video. You see that some of the organoids are non-invasive, some are collectively invading, meaning groups of cells are in a bulky way invading into the matrix, and others are disseminating with lots of single cells. We wanted to understand how this worked, so we cultured organoids from the same tumor and then sorted them by the shape, pooled them by shape, and analyzed and compared every gene these cells expressed, then used engineering mathematics to identify which were the most likely drug targets, and we've identified strategies that are able to block invasion despite this cellular heterogeneity. They're able to block both collective invasion and single cell dissemination. So it gives us a lot of hope that these approaches can be used to find new therapies. What we're doing now is scaling up our ability to work and test new hypotheses. So we're testing which genes and proteins to target at an increasing, I would say, we're accelerating the pace at which we can test new ideas. So as short as 5 years ago, this was really something where we tested one idea at a time. That progressed. 2021, we made major advances in the automation, despite all the constraints in the pandemic. So we can now test 1,000 or more genes or drugs at a time. And so we're able to transition from being predominantly a cancer biology lab to being a lab that both studies cancer biology and tries to apply that knowledge to discover new therapeutic targets. The big ideas here are that tumors build different equipment to perform different tasks in metastasis. I want you to think of this like a triathlon, right? You don't bring the bicycle to swim, right? You don't bring your running shoes in the water, right? There's different tools being used at different stages. We use cutting edge sequencing and mathematical techniques to identify all the changes and then to distinguish which of the changes could be best targeted therapeutically. We then test these ideas in laboratory models of cancer and in fresh patient tumor tissue, and we collaborate with clinicians to translate our discoveries. Where are we going next? We're trying to understand how the immune system limits or sometimes unfortunately accelerates metastasis. We're collaborating with engineers to apply imaging and artificial intelligence to better diagnose and treat metastatic tumors. And we're working with both physicians and surgeons to test new therapies in patients and optimize the use of existing therapies. We're also very importantly partnering with Dr. McConkey and the GBCI to extend our approaches to bladder cancer. And so far, we're seeing exciting evidence that there's similar underlying molecular principles. I want to again thank my laboratory for the very hard work they do to keep this research program going in good times and in bad, and the National Cancer Institute and a series of foundations for the support they provide. All right, thank you, and I welcome any questions you might have.
Fantastic. So we've got a couple already in the Q&A. Let's start with those. So Neil Kurtz has asked, to what extent is the presence of ctDNA a predictive biomarker of future development of identifiable node positive or distant metastatic disease?
So that's not the focus of my research, but I would say in breast cancer, there's evidence that circulating tumor DNA, if it's done correctly, it does provide evidence for the overall tumor burden, right? You can focus the ctDNA analysis on the amount of on specific changes in that patient's tumor. So you'll see before surgery, it's at a high level, it drops after surgery, and it'll tend to stay low if the patient is responding well, if there's little evidence of disease, and can climb back up during recurrence. I'm not expert in how frequently that changes the clinical decision-making.
I'm going to jump. Here's another really good question, Andy. Is there a relationship between the environmental matrix you discussed and the field of epigenetics?
Absolutely. So that's something that's a current focus of the laboratory that I didn't have time to get into. So yes, when those cancer cells are invading, everything they're doing is different, right? Their shape's different, their behavior's different, and we anticipate that is related to epigenetic changes. For those who may not know that term, genetics is about changes in the DNA that are heritable, and epigenetics is changes essentially in everything else other than the sequence of the DNA that are also heritable by cells. So modifications of the DNA or the histones. I have a postdoc and a graduate student working on different aspects of epigenetic regulation that allows cells to turn genes on or off to accelerate or slow down invasion. I think they're deeply.
And the same is true in bladder cancers. There's also a basalization of cells, as Andy pointed out during his talk, at the edges that promote invasion, and some of the same exact genes that he discussed are involved in bladder cancer. So what he's learning about epigenetic regulation in breast cancer and other models is directly applicable to bladder cancer. So Katie Hahn says, great analogies you used. How soon can we expect genetic testing on bladder cancer patients and use that in research? Maybe I'll answer that. So when you talk about genetic testing, a lot of it's already going on. I think the question is whether we're testing the tumor itself, and we're doing that all over the place. So back to Neil's question, we're checking the tumor through biopsies. We sequence biopsies. We also sequence plasma from the blood for circulating tumor DNA. That's probably what most of you have heard about when you hear about liquid biopsies. We're also sequencing tumor DNA in the urine. And our hypothesis, we don't know that this is a fact yet, but it seems to be supported by what we see, is that if there's tumor DNA in the plasma, oftentimes it's a harbinger of metastatic disease. Oftentimes, if we find bladder tumor DNA in the plasma, patients with that ultimately develop metastatic disease later. It's also a way to tell us that we need to add systemic therapy. We need to treat the patient with more than just surgery or local therapy in the bladder. We need to try to eradicate that metastatic disease that we haven't seen. And that's where Andy's research comes in. Because right now, the only tool we really have that does that super effectively and permanently is immunotherapy. But we also know that certain things augment the effects of immunotherapy on metastatic disease. We don't know the rules governing these interactions, and that's why Andy's new research program focusing on how the immune system talks to metastatic cells is so important. Now, genetic research is also focused on hereditary factors, right? So we know now that at least 15% of patients with bladder cancer have hereditary defects in their DNA that predispose them to cancer. We don't know the cause-effect relationships between these effects precisely, but oftentimes what we see is that a patient will have one copy that's defective in, say, the blood cells, and then within the tumor, the second copy is defective in the tumor, which is pretty strong evidence that that particular mutation was selected for, if you will, by the cancer. So ultimately, I personally think that there are going to be a lot more genetic factors that predispose patients to bladder cancer. Right now, we're just looking where we've seen genetic predisposition in other cancer types. But I think as we get more and more sophisticated with our ability to understand what we're looking at, I think genetic sequencing is going to tell us a lot more about which factors promote cancer, and in fact, also which factors might even promote metastasis. So Doug Sharp asks, how are your findings for breast cancer expected to be the same or different in bladder cancer?
So I actually picked the examples I showed in part to highlight things that I would be able to say we've already shown are looking very similar. So as soon as David moved here from MD Anderson, we started talking, started working together, and we have a very open relationship where when someone in his lab wants to learn something, we just literally make room at one of the benches for them to come over and learn it. And that's been going on. And that the student who started that has since graduated. Actually, it's been going on for so long. So we were able to help them figure out how to grow these cells in a very similar 3D environment. And that basic behavior difference that in the collagen one, they take off, and in matrix gel, they really sit still, that seems really conserved in bladder cancer. One step further, this basoluminal phenotype, the cells at the front having this basal differentiation as they invade, also seems conserved in bladder cancer. It seems to be actually, as far as I know, you may know more than me on this, David, but as far as I know, the place where it's most directly conserved is breast and bladder. They seem to really be using not just one or two genes the same, but that whole broad basal molecular program, and using it to collectively invade into the surrounding tissue as a precursor to metastasis. So that looks good. I would say so that was an example where we were leading. An example where we're catching up now to David 10 years ago would be the triple negative breast cancer story I told you about hybrid EMT. So when David came here, he was talking all about how EMT was being used in bladder cancer. And we were like, well, we don't see it, right? We're looking at this luminal breast cancer, we don't see it. So we started getting into triple negative breast cancer. And now we're seeing just what he was seeing in certain subtypes of bladder cancer. So I would say that third story is clearly coming true. And then the fourth one, in terms of, unfortunately, some tumors, especially those that lose P53 and have loss of control of their genome, are very heterogeneous. Different parts of the tumor express different genes, and they behave differently, even in the same microenvironment. I ended on that because I'm worried that that's going to be very similar too. And we already have a new student prepared to directly address that. Actually, I think you already have evidence from that from cell lines.
We have it too with tumors, unfortunately. So Andy's right. So I guess the The thing about this whole process that's a bit frustrating from a therapy point of view is it doesn't seem that the major processes that Andy discussed with you today are necessarily hardwired into the tumor cells. So, for example, for those of you who've been following the development of targeted therapy in bladder cancer, you'll know that activating mutations in FGF receptor 3 identify tumors in patients who will benefit from the drug erdafitinib and more recently pemigatinib and others. Unfortunately, the transitions that Andy showed you today, I think you've noticed by definition that they can be regulated by things like the proteins in the environment, presumably by a lot of other things. And that plasticity means that they can adapt to the pressures that we apply to them. So we do see already evidence for multiple phenotypes, luminal, basal, mesenchymal within a given tumor. And in fact, we're now looking to see whether our therapies, when we apply pressure on patient tumors, whether they push them in one direction or another. So this plasticity, I think, is an important subject of investigation and an important thing to consider with our therapies.
I can actually connect to the questions as well by saying we've got a drug compound library of about 500 compounds that lets us mess with every enzyme involved in epigenetic programming. So it lets everything that's going to read, write, or erase one of those epigenetic marks. And those we've found compounds there that affect the plasticity and block the cells in different states. And so as ideas like that come up, we share them with David. He's able to test them over there. And I hope no one here has experience with any other cancers in their family, but we've definitely shown that key parts of this, not the basal luminal, but key other parts of it are conserved in pancreatic cancer as well. And we're currently partnering with GI surgical oncology to look at adrenal gland, appendix, gastric, small intestine, large intestine cancers. And we're finding, again, there's organ specific features, but there are some common themes there.
Right, and that idea that we might be able to park them in a specific cell state and thereby prevent them from moving around and escaping through plasticity is one concept that we're very enthusiastic about for patients with bladder cancer. So how do chemotherapy and/or immunotherapy drugs and even radiation change how the cells metastasize? Can you explain what that might look like? And thanks for the amazing images.
First of all, you're welcome. So how does therapy change this? So there's a couple of ways that I want you to think about that. So therapy can kill a large fraction of so at the most basic level, let's say we have an effective therapy that kills 90% of the cancer cells. That's going to stress if there's no magic, no cancer stem cells, nothing particularly interesting about the cells that survive. They might just have been by plumbing of the blood vessels, they might just have gotten a lower dose of the drug. Right, that's going to open a large ecological niche for them in the tissue, and it's going to provide a lot of stressors for them. Right, so I want you to think that the first level in which therapy is going to have consequences for cells is by killing the neighbors and providing a lot of danger signals and a lot of dead cell signals. It's going to change the way the surviving cell, the environment around the surviving cells, and it's going to cause them to enter stress states that might be more resistant to further therapy. I would say when you think about chemotherapies in general, for the most part, right, those are either going to work or not work at the cellular level. A given cell will be killed by it or will survive. When you think about targeted therapies, that could kill the cell, that could have no effect on the cell. It could also change the state that the cell is in. So it could be positive change or a negative change. And radiation, I think you want to add on top of that radiation is well known, as are some, but not all chemotherapies. It's well known for stimulating the immune response. Radiation kills cancer cells in a way that exposes their guts pretty successfully to the immune system. So when the right immune cells are there, killing them with radiation can actually have a significant effect of priming the immune system to respond.
Thanks for that, Andy. So Katie, again, are breast and bladder cancer estrogen dependent? I'll take that one. So the estrogen receptor in breast cancer we can consider is substituted by a receptor called PPAR gamma in bladder cancer. Some of the same proteins that help the estrogen receptor, including proteins like GATA3 and FOXA1 also help the PPAR gamma in bladder cancer. We think that there is a role for estrogen and testosterone in bladder cancer that's been established in preclinical models. And we know that women get bladder cancer less often than men. When they do get cancer, it tends to be more aggressive and belongs to the basal subtype. We don't understand that at all. But so we don't think that bladder cancer is estrogen dependent in the same way that breast cancers are. We do think that they're estrogen regulated, and we think that the function of the estrogen receptor in breast cancer might in part be substituted for by another very similar receptor in bladder cancer. Good question. So Neil, why is N1M0 disease stage 3B and N2M0 stage 4? So from a biological perspective, Neil, I'm not sure that I can tell you there is a difference, but from a prognostic point of view, the number of lymph nodes that's positive is associated with your prognosis. So having one positive lymph node gives you a better prognosis than having more than one. With that said, there's a great interest now in using both radiation and surgery to consolidate patients who have oligometastatic disease to lymph nodes. What that means is there's a handful of lymph nodes that's positive. There's also interest in doing that with distant metastases, either with radiation or surgery. And Noah Hahn has kind of pioneered that in our group very successfully in some of his patients. Your second question, which bladder cancer patients with node-positive REM plus disease are candidates for consolidative cystectomy after a favorable response to systemic therapy? That needs a clinical trial. So there's good evidence good anecdotal evidence from some of my colleagues at MD Anderson, led by Nima Navai, that treating the primary tumor can benefit patients who have metastatic disease, as long as they have a beneficial response to systemic therapies. It doesn't have to be a complete response. But since it's only anecdotal evidence at this point, it's not become standard of care. So Nima is trying to lead a clinical trial through the cooperative groups to test that in patients. I would say if you're interested in that, you should talk to your urologist and your medical oncologist about why they or he or she favors one approach versus the other. But there, as far as I know, no strong clinical trial-based evidence to do one versus the other. So Andy, maybe I can ask you a question too, and that is, you told us about the drug screening that you're doing to identify candidate therapeutic targets. Can you elaborate on maybe one or two of those for us to give us some examples of what might be possible in bladder cancer as well?
Sure. So I'll start by elaborating on the epigenetic drug screening, because I think that highlights, there's two strategies where that could benefit. The first is that, so again, epigenetics is a catch-all for anything which is a heritable change, not to the sequence of the DNA. So you can actually make molecular modifications to the DNA, you make molecular modifications to the proteins around which, there are the spools around which the DNA is looped. And so what's exciting there is that the cancer cells seem to need to change their state in order, several times, in fact, in order to complete the stages of metastasis. So you can actually imagine a really different way of thinking about cancer therapy using that epigenetic approach, right? We're not the first, but it's definitely been epigenetic therapies been tested in the clinic for a number of years now. But the way that we think we can contribute is by understanding which of those transitions are critical and which of the transitions that are critical to metastasis are not happening in the adult, right? If you think about an embryo, right? You know, when a woman is pregnant and you've got a baby developing inside of her, lots of things are changing. Lots of developmental programs are being activated and turned off minute by minute, hour by hour, in order to build a brain, a heart, a set of intestines. Whereas when we're adults, change is slower, right? Hair gets thinner, hair gets grayer, we get a little thicker around the middle, the joints stop working as well. But it's not sudden day to day in the same way. And so the hope is that by targeting some of those transitions, there might be very few or no normal cells in the body that urgently need to transition between cell states today and tomorrow, whereas those cancer cells might. The other thing I'll say is that the basal state that they're in while they're traveling is inherently more resistant to chemotherapy. We've been able to do that side by side, measure essentially all the cancer drugs that are commonly used against both basal and luminal cell states. And as soon as they go basal, their sensitivity just evaporates. It goes from being many drugs can kill them to few drugs can kill them. And so if we could kick them out of that basal state, really boring, conventional, familiar drugs that the doctors know how to use might have much higher efficacy at the cellular level. So independent from keeping them from migrating, it might allow you to clear them out wherever they are in the body. So we're very excited about that. I would say the second thing we're doing, it's less bladder cancer specific, but there's a specific clinical decision to treat in GI cancers where they're trying to treat the tumor inside the abdomen. And we're working with Fabian Johnston and John Greer, who are surgeons in the GI surgical oncology division, to try to understand which of those therapies is that they could use is most appropriate across the most patients, disease by disease in the peritoneum. And I think similar approaches could be used in bladder cancer. In fact, David's already using them.
So we have a kind of cancer called plasmacytoid cancers that tend to metastasize through creepy crawling mechanisms into the peritoneal cavity. So anything you learn about the interactions between breast cancer cells and GI cancer cells and the host microenvironment, I think will be relevant to not just bladder cancer, but all the solid tumors that we're trying to treat.
So we know how to grow the cells there, so we should definitely, it sounds like we need to send a student over again.
Absolutely,
It's
absolutely.
very, very recent data, so.
Yeah. And we too are very excited about targeting these chromatin modifying enzymes. So Noah Hahn, if two of his trainees are on right now, Gabrielle and Alyssa, are studying these same classes of drugs for very similar reasons in the lab at the GBCI. So we're getting close to the top of the hour. Are there any other questions from the audience? Well, if not, Andy, I'd like to be the first to thank you very much for a beautiful presentation, super accessible, hit all the high notes, and also for your friendship of the GBCI and your strong collaboration with our group. We look forward to bringing you back again for an update, hopefully as we can talk about all the work we're doing together. And good luck to your program. You're doing the best work I know of across the institution. So keep up the good work. And thanks all of you for joining us today. Don't forget, we've got another one coming up soon. Just check the website, and also the women's program. And Dr. Hahn will have a unique kind of session coming up soon. So stay tuned. Take care, everyone, and we'll see you again very soon.